connected around the cell body. Bipolar neurons are always sensory. They're found in organs for what's called our special senses, which are the ones that you only detect in a certain place in your head. So vision, hearing, smell, taste. Those organs have bipolar neurons to pick up the message and send it toward the brain. Unipolar neurons, okay, if it's only got one, we got to ask, is it an axon or a dendrite? It's an axon, but it doesn't start at the cell body. It just goes right past it. The cell body is stuck off to the side. There are dendrites, but they just come together and start at the axon rather than connecting to the cell body. Unipolar neurons are also sensory. They're used for what's called general senses, where we detect it at various parts of the body, like the sense of touch. So here's one long axon with the cell body off to the side. And then dendrites would come together to start that axon. Questions on structural classification of neurons? They're still going around. So, functional classification, we have sensory, which means they carry messages toward the central nervous system. You might also see them called afferents. Afferent means going into something. Sensory neurons are unipolar or bipolar, depending on which sense they're carrying. Then we have interneurons. This is a contraction of integrative neurons. These are the ones in the CNS. They're the decision makers. They do the integrative information. And then motor neurons. Carry their messages away from the CNS. Where sensory is sometimes called afferent, motor is sometimes called efferent, which means coming out. Think A for arrive, E for exit. We'll use those terms again with the kidney. At that point, it's going to be important to remember. Here, I'm just mentioning them in case you run across them. You can just call them sensory and motor neurons. And then interneurons and motor neurons are multipolar. Questions on functional classification? All right. So that's it for neurons. Our next cell type is neuroglia. There are 6 types of neuroglia. 4 of them are found only in the central nervous system. The other 2 are found only in the peripheral nervous system. Do you notice anything about the names? The peripheral ones are both called something cells and start with S. Okay? So if you see, if it's an S, you know it's peripheral. Schwann cells and satellite cells. The central ones are called astrocytes, oligodendrocytes, microglia and ependymma. Sometimes you'll see these called ependymal cells. So the cells part won't necessarily tell you, but the S will. Astrocytes are the largest and most numerous neuroglia. Astrocyte means star cell. They have a lot of projections like neurons, but instead of axons and dendrites, they're all the same, and a lot of them have these little feet on the ends where they connect to capillaries or neurons. What astrocytes do is maintain a stable internal environment in the nervous tissue. That keeps the conditions right for the neurons to do their job. A major part of controlling the environment is controlling something called the blood-brain barrier. Has anyone heard of that before? Capillaries, the smallest blood vessels that deliver nutrients and pick up wastes, have a certain permeability to them, how easily things can get through them. Around the brain, they're less permeable than anywhere else. That protects the brain from toxins and helps control the content of the fluid around the brain. The astrocytes, with these little feet, are communicating with the capillaries and regulating what gets through. The blood-brain barrier is really helpful to protect your brain. It can be a problem if there's, say, a medication that you want it to act on the brain. You have to be able to get that medication through the blood-brain barrier. An example of this is with Parkinson's. What's usually deficient is a neurotransmitter called dopamine, but they can't just give somebody dopamine and have it get where it needs to go because it won't cross the blood-brain barrier. So they have variants such as levodopa that can get through the blood-brain barrier. This is also where some antihistamines make you drowsy and some don't. The old ones like Benadryl get through the blood-brain barrier, where histamine is also used to keep you awake. So they cancel that out and make you drowsy. Or a lot of the newer ones can't get through, so they are less likely to have that drowsiness factor. Questions on astrocytes or blood-brain barrier? Oligodendrocytes are a cell type that provides myelin. We mentioned before that it's provided by other cells. This is a long name, but we've already seen two of the pieces. What's site? Cell. Dendro we saw in dendrites. What did that mean? Branches. Oligo means a few. So unlike astrocytes and multipolar neurons that have lots of projections, oligodendrocytes have 2 or 3 attached to their cell body. And at the end of each one of those is a roll of myelin that goes around an axon. So oligodendrocytes will be found in white matter of the central nervous system. Microglia, which means little glue. Neuroglia means nerve glue because, glue, like sticking together. Because when they first discovered them, they didn't know all the complex things they did. They thought, oh, this stuff is holding the neurons together. So they called it nerve glue. And these are the little glue. Their job is the same as the macrophages in connective tissue. They fight infection and clean up damage. And they have all these little feathery projections. All those little points can recognize bacteria and things in the tissue. Then the last central ones, ependymma, are like an epithelial tissue for the nervous system, only they're part of the nervous tissue itself rather than a separate attachment. They form sheets like this that line cavities in the central nervous system, called ventricles and canals. These are spaces filled with fluid. Certain areas, the ependymma form a special structure with capillaries where they produce cerebrospinal fluid, which is what fills those cavities. So right here is, let's say it's the central canal of the spinal cord. It's covered by a row of ependyma, or ependymal cells, and then they have some little projections that go into the rest of the nervous tissue. And you can see an astrocyte and a microglia there. Questions on central neuroglia? Here's a little chunk of nervous tissue, putting them all together. And this is just the picture I broke up into smaller pieces to show each kind. This one's a neuron. We can tell in part because it has the myelin around it from the oligodendrocyte. There's a microglia, an astrocyte, another microglia up there, and some ependyma. So this is what you would see in central nervous tissue. We will look at some in lab, but you won't necessarily be able to differentiate neuroglia from each other, just from the neurons. The peripheral neuroglia, Schwann cells and satellite cells. The Schwann cells provide myelin to the peripheral axons, so they have the same job as the oligodendrocytes. Where the oligodendrocytes have a cell body and 2 or 3 myelin sheaths attached to them, each section of myelin is the Schwann cell, and it looks kind of like a sleeping bag wrapped around the axon. Then satellite cells are basically equivalent to astrocytes. There isn't a blood-brain barrier for the peripheral nervous system. I guess we would call it a blood-nerve barrier. They don't need as much careful protection. But there are places where you've got cell bodies of the peripheral neurons clustered together. They need the satellite cells to hold their structure and help control the environment of that bulge. That bulge is called a ganglion. That's the single. Ganglia is the plural. These are places where the cell bodies of the neurons are clustered together. One place that we'll see them in the next chapter is next to the spinal cord, where all of the sensory neurons that are unipolar are coming in. They have these cell bodies sticking off to the side. And instead of them just being lined up somewhere along the nerve, they're all in this one group. So you get this big bulge in the nerve, and that's a ganglion, and it has satellite cells in it, where the Schwann cells will be along the axon for sensory or motor, or even for some interneurons. Questions on neuroglia? One more structural thing to look at is the synapse. We've seen an example of this, the neuromuscular junction. We're going to add a couple of terms. There's a presynaptic cell which is the cell bringing the message to the synapse. This will be the synaptic knob of the presynaptic cell. There's a gap between the cells, and that's the synaptic cleft. And then the cell that receives the message is called the postsynaptic cell. At the neuromuscular junction, the postsynaptic cell is the muscle. When the connections are made within the nervous system, the postsynaptic cell is another neuron, and the synapse will be made along a dendrite or the cell body. Very rarely, but sometimes on an axon. So there would need to be some kind of neurotransmitter released by the presynaptic cell that binds to a receptor on the postsynaptic cell to carry the message. Now, there's two different ways this neurotransmitter could affect the postsynaptic cell. It can excite it or inhibit it. We call that an excitatory or inhibitory stimulus. An excitatory stimulus makes the cell more likely to have an action potential. At the neuromuscular junction, we had excitatory stimuli. The acetylcholine starts an action potential in the muscle cell. An inhibitory stimulus actually quiets down the next cell, makes it less likely to have an action potential. Any given stimulus from one little synapse would be one type or the other, but a single cell could have both types coming into it from different cells. An example of this would be pain sensors in your brain would have the peripheral sensation